Okay, let's dive into this. Welcome to this explainer on the BRCA blueprint and precision DNA repair. We're going to examine one of the most critical, literally life-saving, microscopic rescue missions happening inside your cells right this very second. I want you to picture a totally catastrophic event deep inside the nucleus, like severe trauma to the DNA double helix. We're talking completely severed across both strands, a true double-strand break. This crisis is essentially a blaring 5-alarm emergency threatening the cell's entire genomic integrity. So, can the cell repair this? Just think about the sheer peril in this exact frozen moment. Those broken ends of the DNA are physically drifting apart from each other. It's this absolutely critical biological second where vital genetic information could literally just float away. And fun fact here, your cells actually experience dozens of these severe breaks every single day. Dozens. So if the cell just randomly guesses how to stitch it all back together, it pretty much guarantees a dangerous mutation. But hey, don't panic. Healthy cells possess an incredibly sophisticated protocol to handle exactly this kind of crisis.
Homologous recombination. It's a highly accurate DNA repair pathway coordinated by the BRCA1 and BRCA2 protein complex. And this brilliantly illustrates where the cellular rescue team arrives on the scene. The BRCA1 and BRCA2 proteins rapidly and confidently dock onto the damaged DNA. They act as the absolute crucial site coordinators for this homologous recombination pathway. You see, they don't just haphazardly glue those broken ends back together. Actually, scratch that. It's more like they secure the whole perimeter, recruit nearby repair proteins, and expertly manage what is honestly one of the most precise biological operations in nature.
Step 1. Align an identical, healthy DNA molecule as a template. Step 2. Perfectly copy the missing DNA sequence to reconnect the broken strands. Let's move to the actual mechanics of this and see how this builds because it's absolutely ingenious. Think about it. How do you fix a torn page in a book without losing a single vital word? Well, first, the cell aligns a second, perfectly identical, healthy DNA molecule, usually a sister chromatid, right beside the damaged one to act as a pristine template. Then, this amazing transfer of information begins. The missing sequence is perfectly copied from the healthy template to bridge the gap. Thanks to the BRCA complex, the cell never, ever guesses. It uses the master copy to patch the break with flawless accuracy.
DNA restored, genome stable. So, the crucial point is the ultimate impact here. The result is just a biological masterpiece. The broken strands are fully reconnected, and the DNA is restored. When these BRCA proteins function normally, even these highly dangerous double-strand breaks are flawlessly resolved. The cell survives, it continues to divide normally without passing on any fatal errors, and it totally averts a major genetic catastrophe. Your biological blueprint is kept perfectly intact. But what if BRCA is missing? As we wrap up this explainer, I want you to consider a pretty dramatic shift in this scenario. What actually happens when this crucial BRCA setup fails, when that rescue team never arrives to coordinate the repair? Well, those severe breaks either just remain wide open, or they get frantically, desperately patched with really dangerous genetic errors. And this leaves us with a truly provocative reality. These unrepaired breaks, born from a missing defense system, are exactly where cancer cells become incredibly vulnerable. It really makes you wonder, doesn't it? How can modern, targeted medicine actually use that very missing system as a weapon against the disease itself?